All right, good afternoon, everybody, and welcome to the Genomics England Research Seminar. So my name's Matt Brown. I'm the Chief Scientific Officer for Genomics England, and I'm going to be just hosting this afternoon's session. First up, just to notify everybody that we're recording this seminar and that it will be available on YouTube after the event. So just to mention, we have 1 hour for the seminar now. There's going to be questions and answers after each speaker, and if you can put your questions in the designated question and answer feature rather than the general chat, or raise a hand and I'll come to you if I can. So just very briefly, we have Dr. Daniel Greene from New York, Mount Sinai via University of Cambridge, and Rebecca Tooze, who's a PhD student at University of Oxford. So we've got the two blues lined up, ready to roll, and a fantastic afternoon of rare disease genetics that we should have in front of us. Next slide, please. So just to flag, whilst I've got everybody's attention, we now have a date for the Genomics England Research Summit. It's going to be on the 19th of September. The three main tracks are going to be rare diseases, cancer genomics, and emerging technologies. For those of you who were able to attend the meeting last year, I think it was absolutely fabulous. It was certainly, in terms of meetings attended, the highlight of the year as far as I was concerned. We're going to have oral and poster presentations, and so we're really keen to get abstracts in from both internal Genomics England researchers and also from external researchers working with Genomics England through GSIPs or Discovery Forum, whatever. So if you could send your submissions to the email address that's here, events@genomicsengland.co.uk. The final deadline for these is actually not that far off, so you need to get your skates on. It's 5th of May. So look forward to receiving your abstracts for that. And so just the last slide, if we could, is that the next meeting, the next research seminar that we'll be doing is at the end of May. And for that, we're going to have a showcase of early career researchers. I won't go through all of their topics, but we've got 5 excellent early career researcher presentations for that. It should be a really exciting day. And I really appreciate the early career researchers putting themselves out in front of a big audience like this to do what should be a fascinating afternoon. All right, now over to our first speaker. So our first speaker is Assistant Professor Daniel Greene. As I mentioned, previously of University of Cambridge. I won't read through his entire CV or bio for you, but other than to say that he's a mathematician who's worked extensively in genomics and in biostatistics in rare diseases, working with Professor Ernest Turo. And most recently, in fact, this month, he's published a paper in Nature Medicine, which works extensively on rare disease genomics and novel methods for identifying previously unidentified genetic variants in our rare disease cases. He's recently been appointed to the faculty of Mount Sinai School of Medicine, in the Icahn School of Medicine, rather, in New York. So congratulations to you for that appointment. And I really look forward to your presentation now. Over to you, Daniel.
Hello, everybody. Thank you very much for tuning in. My name is Daniel, and I'm very pleased to speak to you today about my recent research project, the genetic association analysis of 77,000 genomes reveals rare disease etiologies, which is, of course, based on the 100,000 Genomes Project data. So as background, as researchers who are working on large whole genome sequencing studies who are interested in novel etiologies, the starting point for the kinds of cohort-wide analyses that we want to do are the merged VCFs. So in a study such as the 100,000 Genomes Project, these merged VCF files would contain hundreds of millions of variants corresponding to hundreds of millions of rows, tens of thousands of samples, so tens of thousands of columns. And so obviously working with such large files as these is very difficult to create them and update them and also read them, requiring a lot of compute power. Additionally, the phenotype data is not stored in these files. So if you want to query the data in a way that depends on the genotypes, the variant annotations, and the phenotypes in one go, that requires writing code, certainly writing more code than should the data be contained in a database. Furthermore, as many rare diseases are caused by individual rare variants or by pairs acting dominantly or by pairs of rare variants acting recessively, we are particularly interested in these rare variants. And what we found is that by restricting allele frequency to under 1 in 1,000 across all major populations, we can reduce the number of non-homozygous reference genotypes that we look at by 99%. So this plot at the bottom on the left shows the proportion of such non-homozygous reference genotypes that we would retain by applying these criteria, and on the right, the raw number of them across the major ancestry groups that are represented in the 100,000 Genomes Project. So as you can see, ranging from about 20,000 rares per European sample to about 40,000 for an African sample. And so we developed a set of tools for importing, storing, and processing this data in a relational database schema, which we call Reservoir. And this diagram shows a sort of simplified view of the major tables which are used in the Reservoir. And so we have here, you can see the variant table, which contains a variant ID called the RSVR ID, and then annotation from large files. So as examples, CAD Fred scores and frequency information from Nomad. A table of consequences, which links variant transcript pairs with consequences from the sequence ontology, and a table of genotypes, which links the RSVR ID, variant IDs with sample IDs, and the genotypes. So this is a sparse table, so we don't store the non-homozygous reference genotypes. So the genotype could be either 1 or 2, depending on the number of alleles for a particular variant. So these black arrows indicate foreign keys. So these tables are linked together. So there's a table of samples, which contains statistics for each sample, a table of case sets. So this will link particular disease diagnoses to samples, and a table of standard phenotypes. So for example, HPO and ICD-10 would go in this table, and then they would be linked to samples through a link table. So you'll notice that the tables containing variant IDs all have this column, the RSVR ID. The RSVR ID is what is used in Reservoir to encode a variant. And so it's a 64-bit integer, which encodes the chromosome with 5 bits, the position with 28 bits, the length of the reference allele, the length of an alternate allele, and then up to 9 bases. So by default, we use the first 5 and last 4 bases in the alternate allele. And so we encode variants in this way, so that, and a point to make here is that the RSVR ID is increasing with variant position. So when these tables are indexed on RSVR ID, you can do very fast lookups based on genomic position without having to link to join to a table of positional information, but also whilst keeping the structure of the database simple, using just this single column. So 99.3% of variants that you encounter can be encoded losslessly based on Nomad. And then also based on Nomad, 99.8% can be represented uniquely within Nomad by encoding variants in this way. Another aspect of the structure of the database is the consequence ID, which is used to annotate these overlapping variant transcript pairs. So the consequence ID is again a 64-bit integer, such that each bit represents a particular term in the sequence ontology. So there are fewer than 64 such sequence ontology terms which we can annotate variants with, and so we can therefore sort of efficiently encode all of the possible consequences a variant might have by using one of these IDs. Furthermore, the bits within the consequence ID are ordered in increasing severity, which avails queries which threshold on the severity or order by the severity of consequences to be written neatly. So the process for building a reservoir is done in stages, starting from either a set of single GVCFs. So that's the process sketched here on the left, or from a merged VCF, which is here shown with the blue arrows. This process takes place by using the set of standard Unix tools, including BCF tools, and also with the complementary software package that we wrote for managing reservoir, which is the RSVR package, which is available on GitHub. Using these tools, you can import a set of GVCFs or a merge VCF into a reservoir. So these two separate pipelines unify at the point at which you find the set of rare genotypes from aggregating across these VCF files. A key step in both processes is to filter out the variants which are predicted to be common in Nomad in any of the major populations. And so once that's done, the processes become the same, and you then use the sub-program of RSVR seekeffects in order to annotate variants with sequence ontology terms and optionally filtering, and also the RSVR anno package, which can extract information from these large annotation files, for example, CAD and Nomad. So we executed this for the merged VCFs in the 100,000 Genomes Project main program, pulling out variants in exonic regions according to ensemble build 104. We applied it in parallel over 1,000 merged VCF chunks, and in total, I think it took between 2,000 and 3,000 CPU hours. The main bottleneck of the process being the actual reading of the VCFs. So what we're left with is a database which contained 60 million genotypes, 14 million variants, 12 million transcript consequences observed in these 77,000 samples. We also imported the 216,000 HPO terms from release version 14. And in total, we're left with this quite compact database, which is 5.5 gigabytes in total size, so obviously a lot smaller than the original files. So this diagram on the right shows the breakdown, the data that is stored that annotates the variants in reservoir. So you can see arranged in terms of the sequence ontology with the annotation that's available in reservoir, so the CAD scores and the nomad allele frequency classes. So now that we have this genetic data in place in the database, we want to systematically analyze the disease categorizations that have been availed through the research environment. So you may know that the disease classifications in the 100,000 Genomes project are arranged in this hierarchy where participants were assigned one of 220 specific diseases, which were arranged into 88 disease subgroups, which were arranged into these 20 disease groups. So in order to perform the association analyses, we need to establish sets of cases and controls. And so we decided to include both the specific diseases and the disease subgroups as case sets or disease classes. So this allows, to an extent, for variable disease presentation, but for a given etiology to be taken into account. And so this diagram on the left shows the sizes of the case sets which we analyzed, including the disease subgroups in blue, specific diseases in red, organized by the disease group. And so in total, we were left with 269 unique sets of individuals. On the right here, we can see an example of the disease classes which are within one disease group for cardiovascular disorders. So the method that we wished to apply to analyze these case sets was the BeviMed association analysis, which we published in the American Journal of Human Genetics in 2017. BeviMed is a statistical method which computes a probability of association between the disease status, a case control status, and rare variation observed within a gene or a genomic locus. So it works by comparing models for dominant and recessive inheritance using different allele frequency thresholds, up to 1 in 10,000 for dominant, 1 in 1,000 for recessive, and across different classes of variant, depending on the predicted consequences. So in this analysis, we compared 5' UTR variants with moderate impact variants. So that's having a missense or more severe consequence, or loss of function, high impact variants. And so in this way, BeviMed enables you to infer the class of variants responsible and also a mode of inheritance, and gives you a probability of association overall. We applied it to all of the 269 classes for all of the 19,663 protein coding genes in Ensemble 104. And we retained the associations which had a probability of over 0.95. So we found 260 such associations. 241 of these were known by according to PanelApp, by matching the disease classes with panels in PanelApp. So PanelApp is, for those of you who don't know, an expert curated database of disease gene associations with different levels of evidence, with green for strong evidence, amber for medium evidence, and red for weak evidence. And so with all of our 260 associations, we colored the association according to the level of PanelApp evidence for which we could obtain a match between the disease class and the panel. So we have 241 of these associations already seen in PanelApp, and we also have 19 novel associations, which are shown in grey. So here we've arranged the results by disease group, and I hope you can see that some of the associations are shown with a circle, and some of them are shown with a square. The circles are with specific diseases, and the squares are with disease subgroups. And so about 20% of these results are with squares, so with the disease subgroups, which demonstrates that we are increasing power by including the disease subgroups as case sets in order to account for this phenotype heterogeneity within etiology, as we set out to. In order to follow up some of these 19 novel associations, we then arranged them by additional sources of evidence, including the evidence of purifying selection in NOMAD, evidence of co-segregation between implicated variants and disease status within the extended pedigrees of cases for these results, and then also by conducting a literature review to look at the biological functional evidence which could support the role of these particular genes in the diseases for which they were associated. So we shortlisted three results which had a score of which found positive evidence in all three of those categories. So one of these associations was between ERG and primary lymphedema. So here the association was based on the observation of frameshift variants in 3 affected families, which you can see in panel A. There was a 4th family that was recruited for an unrelated condition, but upon which also carried a frameshift mutation. However, on chart review of this individual, they also exhibited symptoms of lymphedema, so that provided internal replication of the result. We did observe. So you can see here in the pedigrees the empty symbols show for when we don't know what the genotype is, but we observed that the affected father of one of the cases, who was actually called homozygous reference. When we actually looked at the reads supporting that position, we did find that 2 of the 48 reads actually supported that variant, and therefore this would suggest that this person was actually mosaic for this variant, and it would go some way maybe to explaining why the actual phenotype for the affected father was milder than his affected daughter. Because 2 of these variants occur in the last exon of ERG, they would putatively avoid nonsense mediated decay, and hence we used immunofluorescence in order to study the protein in the cell. So in HEC293 cells, which don't endogenously express ERG, we used overexpression of ERG cDNA in order to study this, and so we did this for wild type and also wild type ERG and each mutant, and you can see from the diagrams that in the mutants the ERG localizes outside the nucleus, which would suggest that the mechanism of disease could be a mixture of loss of function and also mislocalization. The second association, which we
Daniel, just to flag that you're at you've got about 7 or 8 minutes to go until the end, including questions.
Yep. Okay.
Okay.
So the second, thanks very much. The second result that we followed up was between, association was between PMET-A1 and a disease label, familial thoracic aortic aneurysm disease. And so we observed the 3 families with the same frameshift variant were driving the association, and we then found in 3 different replication cohorts, including three individuals from Japanese ancestry with the same variant, and then 2 additional individuals from families with a different variant, the same exact phenotype. So when we looked closer at the phenotypes, we observed that these skeletal phenotypes were enriched amongst these PMET-A1 variant carriers in comparison with thoracic aortic aneurysm disease as a whole. The people resembled the phenotypes of Loeys-Dietz syndrome. And indeed, when we when we selected the protein-protein interactions from StringDB, which are shown here in panel C on the right, here we're showing the genes which are associated with familial, on the familial aortic aneurysm panel, with along with PMET-A1, and we've colored the genes which are associated with Loeys-Dietz syndrome in blue. And as you can see, PMET-A1 interacts with these other genes in the LDS sort of network. The final association which we followed up was between GPR156 and congenital hearing loss. The association was driven by variants in 2 recessive pedigrees, and a third was found through gene matcher. So you can see these pedigrees here on the left. We found that there was a recent paper implicating GPR156 in stereocilia function, and to help interpret this, we looked at the log fold gene expression between hair cells and surrounding cells in mice. So we compared 3 different sets of genes, genes that were not, so in gray, we've got the genes that were not homologs of genes that are implicated in hearing loss. On the hearing loss gene panel in panel app, and then here we've got the genes that are homologs of genes on the hearing loss gene panel, either with or without a stereocilia Go term annotation. And so you can see that clearly the stereocilia annotated genes exhibit higher fold change in expression in hair cells, and GPR156 expressions sits very sort of comfortably in this distribution, sort of adding weight to our theory. And so again, because of the 2 of these variants being in the last exon and then putatively avoiding nonsense mediated decay, we wanted to study the expression. And so we, in order to help validate this, we transfected COS7 cells with wild type and mutant DNA and measured the expression, and observed that the expression was markedly reduced in the mutants. I'd just like to conclude by thanking all of my collaborators who made this possible, in particular Ernest Turow, Kathleen Friesen, and Andrew Mumford. It was very interesting to collaborate with people from all over the globe. So yeah, thank you very much for listening, and I'll be pleased to take any questions.
Okay, we have a couple of minutes for questions, but Daniel, if it's okay, there are probably more questions than we've got time for you to actually answer. Okay. So if you could perhaps answer some of the questions through the chat, that would be great. I'm going to take chairman's prerogative and just ask the first one. As a common disease researcher who's interested in, of course, the common disease field is really interested in rare variants causing common diseases. Have you had a go to see whether this has actually got any utility in trying to identify rare variant associations with common diseases?
Yes, we can pick up some associations. So we looked at we just looked at a few examples from UK Biobank. So we can pick up some associations, yes, although I haven't compared, I haven't done a systematic comparison of using this approach with like common disease methods, but it is possible to pick up signal from some signals from just the rares for some diseases. Okay.
And interestingly, just right now, my access to the questions and answers has actually completely disappeared. So I'm not actually able to see what questions people have asked. And I'm going to ask, if people have got a question, can you put your hand up as well? Because that way I can ask you to directly ask the question. Or better so there was a question in the chat that I remember that had to do with basically the availability of the CHQ ID and the RSVR ID data sets, and whether they're going to be publicly available, or where they might be publicly available.
Well, the software is freely available on GitHub, which you can find through. So this talk is really based on the paper. So the paper links you to the software. So and I mean, reservoir is very small, and so in this instance, for the 100,000 genomes, people would be able to like build their own version, add their own tables. So yes, it's certainly available.
Okay, all right. Shamima Rahman's got a question. Over to you.
Hi, thanks. Really fascinating work. Question I had is: it looked as though there was a case of phenylketonuria that you picked up using your methods, and that's really interesting because of actionability. Had this case not had newborn screening? Do you know anything about it?
Sorry, I couldn't quite hear that.
It looked to me, unless I was reading your figure incorrectly, that there was a case of phenylketonuria there in the metabolic disorders, the PAH. And just because that's a potentially treatable condition, and it's one that we do newborn screening for, and I thought that Genomics England would be interested with the planned newborn genomes project coming up. A bit further to the right.
Well, presumably yes. These are all diagnoses, not just new diagnoses.
Interestingly, as we describe in the paper, the one of the tables in Genomics England, that's available in the research environment, the GMC exit questionnaire table, which has diagnoses in it. Not all of those people are. We have plenty of people who have implicated variants from this analysis who are not in that table. I guess one remark like you could make is that this kind of analysis could help sort of support clinical diagnoses.
Yes, and as Matt says, it's possible it's someone who was known to have PKU that was in the study for a different reason. I guess we just don't know. Thank you.
Yes, yeah.
Okay.
I don't know how up to date that table is, or whether it gets updated from time to time, but it's certainly interesting.
All right. Daniel, we might pause at that point and just say thank you very much. If you wouldn't mind answering the questions in the Q&A just by typing some answers, that'd be greatly appreciated, I'm sure, by those who did ask. So congratulations on a fantastic piece of work, and I look forward to seeing how that develops.
Great, thank you very much.
No worries, thank you. All right, so we might move over now to Rebecca Tooze, who's going to basically, Rebecca is a PhD student at University of Oxford, working with Professors Andrew Wilkie and Stephen Twigg in clinical genetics group, and is going to talk with us about clinical genomic sequencing for the diagnosis of rare diseases with a particular focus on craniosynostosis, I guess. Over to you, Rebecca.
Brilliant, thank you. And I hope everyone can see my screen there okay. So thanks so much for giving me the opportunity to present my work today. As mentioned, I'm a PhD student in the clinical genetics group based at the University of Oxford. And we're primarily interested in the genetics of a rare disease known as craniosynostosis. So the talk that I'm going to give today is an update of the genes associated with this disorder, and then the need for the expansion of the current diagnostic grade gene panels, specifically PanelApp, which has just been discussed. And sort of the need to update this in line with sort of future research. So to start with some background, craniosynostosis is a rare congenital disorder. It's characterized by the premature fusion of the gaps in our main skull plates, known as sutures, which you can see in this image on the right-hand side. We have 6 main sutures, which essentially allow space for the brain to grow during early development, and room for the head to pass through the birth canal. These sutures typically remain open until early adulthood, so sort of early 20s, with the exception of the metopic suture, which fuses during the first year of life. So craniosynostosis prevents growth of the bones in a direction that's perpendicular to the suture itself. This can cause compensatory overgrowth of the remaining open sutures, which leads to the gradual dysmorphism of the skull, which is sort of typically characteristic depending on which suture has fused, as you can see in this image. You can get fusion of these sutures in isolation, so this we refer to as non-syndromic craniosynostosis, or this can be accompanied by additional clinical features, something like hearing or sight abnormalities or intellectual disability. And this, when you get craniosynostosis accompanied by these clinical features, we refer to that as syndromic. So there are both environmental and genetic causes, and this is data taken from an Oxford cohort of 666 individuals. And from this group, a solely environmental cause was identified in about 2.5% of patients. And this is things such as exposure to smoking or alcohol intake during pregnancy, or certain medications, alongside sort of decreased space inside the uterus from twins or reduced amniotic fluid. However, as a sort of genetics lab, we're obviously interested in the genetic aspect of craniosynostosis, and in the same cohort, you can typically identify positive genetic diagnosis in around 25% of the individuals, largely through identifying mutations in just 7 genes or chromosomal abnormalities. But this does leave quite a large portion of the cohort currently unsolved. And this is where we've been using some of the data generated from the UK 100,000 Genome Project to try and identify new disease associations for craniosynostosis, or to try and uncover some of the harder to identify genetic diagnoses in some of these core genes. So in addition to those 7 core genes I've just mentioned, craniosynostosis is also characterized by this long tail of rarer genetic diagnoses. And by this, I mean sort of 2 or a few families that have mutation in a given gene, which is causally associated with the craniosynostosis, but isn't recurrently mutated in lots of different families. So in the same cohort I just mentioned, 3.5% of that cohort actually harbored pathogenic variants in 20 or more rarely mutated genes. And this is really important as in a diagnostic setting where patients are sort of typically screened for variants in panels of genes, and that panel must be reflective of the heterogeneous nature of the disorder. So for craniosynostosis, it's particularly key that sort of all of these longer term rare genetic diagnoses are included in panel-based sequencing of patients. So one such panel used is PanelApp. So as sort of previously just mentioned in the prior talk, PanelApp is an expert-led knowledge base that aims to establish a consensus diagnostic gene list for all the rare disease categories within the 100,000 Genome Project. So for us, there's a panel for craniosynostosis. Each panel includes a traffic light coded system based on the level of evidence for gene disease association. So for green genes, these are ones with high level evidence. There must be 3 or more independent families with a congruent phenotype. So in our case, craniosynostosis. There must be a plausible disease causing variant affecting an interpretable region of the gene, for example, a functional domain. Or if there are 2 or more independent families, there must be additional convincing bioinformatic or functional evidence. So something like the sort of mutation of a specific gene in an animal model, which shows the phenotype of interest. So in addition to that, in PanelApp, there are amber and red genes. So these are the ones with moderate or insufficient evidence for gene disease association, respectively. And this could be that variants are identified in 1 or 2 families, but for which additional cases are needed to establish disease causation. So encompassing this longer tail of rare genetic diagnoses, the PanelApp for craniosynostosis at this point includes 65 green genes, 13 genes that are amber, and 50 red genes. However, the last systematic update to this list was actually in 2019, suggesting that it's been quite a long period where there could have been sort of new discoveries that would promote the status of some of these amber or red genes to green, but these are sort of yet to be included in the latest PanelApp releases. And this is important for sort of routine diagnostic settings where they're mandated to review variants identified in the green genes, but not necessarily the amber or red. And if these sort of the latest PanelApp release doesn't reflect the latest knowledge in the field, then some of the sort of card-carrying genetic diagnoses may be routinely missed. So to provide a sort of quick example of the scale of this issue and the importance of updating these panels, I'm just going to mention a paper that we published a couple of years ago now in Genetics and Medicine. So at that point, there were 114 families enrolled in the 100,000 Genome Project with either complex or familial craniosynostosis. So by complex, I mean they either have multi-suture fusion or are syndromic. And this sort of encompasses the group of patients with craniosynostosis who more likely have a genetic cause to their diagnoses. So at this point, we identified 34 patients in which we would consider having a positive genetic diagnosis. And out of those, 18 of them were missed by the current triaged or NHS diagnostic bioinformatic pipelines. So they were missed for a variety of reasons, and I'm not going to discuss all of these in detail, but just to mention, obviously de novo mutation was one of the biggest ways in which you could pick up some of these missing diagnoses, and this was the screening de novo mutations in any gene, so not just those listed in PanelApp. But importantly, 33% of the missing diagnoses were actually identified in green genes, or those that were considered green. 4 of these in that sort of green segment here were missing indels, because the indel caller at the time filtered out many of these variants based on quality metrics, but has actually since been updated, so should now identify many of those missing indels. And the last 2 identified in green genes were actually 2 genes, which are some of the most commonly mutated in patients with craniosynostosis, but both these were complex copy number variants, which you wouldn't expect would be difficult to identify in a routine diagnostic setting. But importantly, and this will sort of form the focus of the remainder of the talk, there were a subset of variants missed identified in amber or red genes at the point that the patient was recruited to the programme. And this doesn't take into account any sort of recent discoveries that perhaps indicated that genes should have been prioritised more highly. Incidentally, SOX6, which you can see here, is also currently listed as an amber gene, but these 2 variants weren't included in that analysis of missed amber genes because they contributed to the original discovery cohort associating SOX6 variants with neurodevelopmental delay and craniosynostosis. And although this is a small cohort, considering it's sort of only 114 families, you can imagine that there's sort of 100,000 individuals enrolled in this program. And if 20% of the diagnoses are missed because that gene is considered amber or red, when perhaps it should be considered green, that's a considerable number of families who are not receiving a genetic diagnosis. So because of this, we aim to analyse all exome, genome and panel-based analyses of patients with craniosynostosis, encompassing a period from 2018 to 2023, which was hopefully aiming to address this knowledge gap where PanelApp hasn't been updated since 2019. And just to note, this was recently published in Journal Genes back in March this year. The paper's listed on the right-hand side there if you're interested in looking at this further. The sort of papers that were included in this analysis, we identified 14, which are just listed on this slide here. From these papers, I tallied the number of variants identified in genes listed as green, amber or red for PanelApp at the time. And this was considering the version 3 data release for craniosynostosis. As you'd perhaps expect, the majority of pathogenic variants were identified in green genes because this sort of made up the majority of genes included in these panel-based analyses of patients. But there were a significant number of variants which intersected amber or red genes and perhaps could provide evidence to promote the status of those genes. Alongside this subset of 14 papers, I also screened any single case report across any time period to try and identify any additional cases to either promote the status of a current amber or red gene or establish a new gene that hadn't currently been listed in PanelApp at the time. Also in the analysis, you'll see my surname listed down here. I included a resequencing project, which I've been undertaking over the last year or so. And this aimed to sequence 617 individuals for mutations in 42 genes. And as you can see, this is the sort of largest cohort of patients with unsolved craniosynostosis included in sort of all the papers that I analysed. So out of those 617 individuals, I included a sort of wide cohort of craniosynostosis diagnoses, including both non-syndromic, that's what the NS stands for, and syndromic cases, which is the S you can see there, and then considering a sort of all sutures fused, so unicoronal, bicoronal, sagittal, unilambdoid, bilambdoid, metopic, and multi-suture synostosis. So collectively, that made up a sample size of 617. And then, as I say, we screened them for variants in 42 genes. So this 42 gene panel included a subset of 7 genes that were identified as candidates across my PhD, either from the analysis of the 100,000 Genome Project or our in-house database. And the sort of point of this was to try and identify additional cases to sort of establish the role of those genes in craniosynostosis. An additional subset of just over 20 genes were those in which there's sort of more than one reported case, but perhaps for which the association with craniosynostosis is less clear, or for which there is a known role for that gene within craniosynostosis, but for which we aim to establish the frequency of mutation in an independent cohort. And then the last subset of genes included were those in which there's a reported constitutive or loss of function animal model, which presents with craniosynostosis. So considering that, I identified 4 variants in green genes. One of these was a variant in MSX2, which is one of the first genes to be associated with craniosynostosis, and the individual harbored a pathogenic variant at a recurrently mutated position. And then in addition, I also identified 3 variants in ALX4, which is another member of the homobox class of transcription factors. However, as sort of I mentioned, the priority was the analysis of amber or red genes, so I'm not actually going to discuss those ones in any further detail. So in addition, I identified 6 variants in amber genes, 3 different amber genes. So 2 of these were variants in SOX6. One of them was a variant in SMAD3, and then we also identified 3 variants in a gene called PRRX1. So SOX6 was published a couple of years ago. This was a cohort of 19 individuals with neurodevelopmental syndrome, and 2 of these included craniosynostosis, which were the original 2 individuals that I mentioned from the analysis of the 100,000 Genome Project. But importantly, all of these have loss of function variants in SOC6. And within the resequencing analysis, I identified 2 individuals from 2 different families, both with loss of function variants, and one of these was identified to be de novo. In addition, the SMAD3 variant resulted in a missense substitution at the canonical splice site between exon 1 and intron 1. Additional functional analyses that I've been undertaking has shown the complete loss of 1 functional copy of SMAD3 in this individual, which is sufficient to give it diagnostic status. However, the sort of other reports in literature of variants in SMAD3 and association with craniosynostosis are less clear. So currently there's not enough cases of variants in SMAD3 and craniosynostosis to promote this gene in status from amber to green. However, the findings of variants in SOC6 and PRX1, which I'm going to discuss a bit further, have been enough to elevate the status of both these genes from amber to green. So as I mentioned, PRX1, this is the one I've been doing sort of additional functional work, so I thought I would discuss this one a bit more detail. So for background, before this point, PRX1 was considered an amber gene because of its role in sort of a general or more severe craniofacial abnormality known as agnathia ichthyopathy. But until this point, its role within craniosynostosis was less clear. It is a transcription factor, meaning that it obviously enters the nucleus of cells to bind DNA and switch on transcription of other target genes, and specifically has been shown to be very highly expressed during early limb bud formation and craniofacial development. So on the right-hand side, you'll see an image here from the mouse coronal suture at embryonic day E15.5 with the frontal and parietal bone that overlap at the coronal suture, they're stained in green, and PRX1 stained in pink, which is really highly expressed within the sutural mesenchyme there. So across the course of my PhD, and actually before I started the project as well, there have been a number of patients described with craniosynostosis and variants in PRX1. All of them predicted to result in loss of function of the variant allele. The ones that I've highlighted here with the arrow are the ones that were identified through my resequencing analyses. So one whole gene deletion, which actually encompassed 10.5 megabases, but one of those genes was PRX1. In addition to a deletion of exons 2 to 5 and a loss of a start codon in another individual. But collectively, as you can see, has a considerable number of patients with craniosynostosis and reported variants in PRX1, which sort of far exceeds the threshold 3 independent families required for this gene to be green on panel lab. However, one sort of more complicated factor relating to this work is that actually many of the variants which we would consider as likely pathogenic were actually inherited from unaffected family members, which you can see on these pedigrees on the left-hand side here. And there were only 3 families in which we identified a de novo variant, leading to an estimated penetrance of 12.5%. So because of this, we wanted to make absolutely sure that the variants we identified in these patients were pathogenic. And so to do so, we wanted to do some additional functional work to sort of prove what was happening here. And this was particularly important for the missense variants identified in these patients. So as I mentioned earlier, PRX1 is a transcription factor and it localizes to the nucleus. To do so, it harbors 2 nuclear localization sequences at the N and C terminus of the functionally relevant domain known as the homeodomain, which incidentally intersect a number of the variants identified in patients with craniosynostosis. So the question we wanted to ask is whether a mutation of any of these positions resulted in the abnormal nuclear localization of the mutant protein. So as you can see here, this is an immunofluorescence analysis. You've got PRX1 here stained in green, the nucleus stained in blue, and the surrounding cell cytoplasm stained in red. In the left-hand panel, you're seeing the wild type or normal protein, which as expected in the majority of cells screened did localize to the nucleus. In the next 2 panels, these are 2 controls that were included in the analyses. So the D54A variant was the only variant identified outside of the homeodomain, and we anticipated again that that would localize to the nucleus. And then on the right-hand panel, this was included as, again, a control. So this is a known benign polymorphism within the homeodomain. And again, as expected, the majority of cells screened, this did localize to the nucleus. In contrast, missense substitutions identified in patients with craniosynostosis resulted in a very different phenotype, wherein the majority of cells screened, the protein localized to the cytoplasm, preventing PRX1 from performing its normal function. So in total, with this mechanistic evidence, alongside the vast number of patients which we've identified with variants in PRX1, this is sort of substantial evidence to promote the status of PRX1 from amber to green. So in total, considering the sort of exome, genome and panel-based analyses of patients with craniosynostosis, alongside work from my sequencing analysis and any parallel studies I've been undertaking, I've been able to identify 16 genes, which should be newly considered as green genes on PanelApp, increasing the overall number of green genes by 25%. And this should have a very significant positive contribution to the total number of diagnoses identified in patients with craniosynostosis who are newly recruited to the NHS Genomic Medicine Service going forward. So just to point out, only 2 of these genes identified out of the 16 we would consider as core craniosynostosis genes. So by this, I mean that mutation of that gene typically presents in craniosynostosis. And for the remaining 14 genes, we would consider that craniosynostosis is the less frequent feature of variants in that gene. So things like ARID1B and BCL11B on the previous slide are green genes for intellectual disability. So that's the sort of predominant phenotype in those cases. In the paper, and you can read about this if you're interested after, we sort of broadly classified each of those 14 genes into 5 categories of pathophysiological function. The biggest category of those being regulators of self-fate and differentiation, so controlling the turnover of cells within the sutural mesenchyme, so controlling whether they become bone-forming cells or stay within the sort of stem cell population within the suture. And then last, we supported any likely pathophysiological function with data, single cell data taken from the mouse corneal suture at E15.5 to E17.5. And this is data done by our lab, which was also previously published, identifying sort of the likely or where these cells are being expressed within the mouse corneal suture. So on the left-hand side, ADAM TSL4 is a regulator of the extracellular matrix. And quite nicely, we found that this was being expressed within the sort of ectocranial cluster of the mouse corneal suture. BCL11B is a regulator of cell proliferation and differentiation. We specifically found this enriched within the osteoprogenitor cell populations, which is consistent with previous mouse studies that identified an essential role for BCL11B in suture biogenesis. And then last, as I've just discussed with PRX1, this is a transcription factor, which is really widely expressed in sort of all cell types, which we also identified within the corneal suture there itself. So to conclude, we've provided the first systematic update to genes that are associated with craniosynostosis since 2019, identifying 16 genes, which should now be considered green genes, which should hopefully facilitate routine screening of variants in these genes in newly recruited patients to the NHS Genomic Medicine Service. These have been updated now in PanelApp, but unfortunately the review process is quite slow. So if you were to look today, I think you'll find sort of most of the 16 genes I've just discussed would still be considered either amber or red. And I've just also put the paper there at the bottom again in case anyone is interested. So thank you to everyone involved, particularly mentioned to my 2 supervisors, Andrew Wilkie and Stephen Twigg, alongside 2 other members of my lab, who have made very important contributions to the total uplift in patients with a genetic diagnosis and craniosynostosis within the 100,000 Genome Project. So thank you very much for listening. If you have any questions, I'd be happy to answer them.
Okay, thanks very much, Rebecca. We've got time for 1 or 2 questions if you put them in the chat. Just briefly, just to comment that to thank you for your role in updating PanelApp, which was most recently updated in March this year. And so there is a continual update process about it that has to cover all diseases and only a small team to do it. We really value the contribution of GSIP and academic contributors such as yourself and Professor Wilkie's group. So can I just ask at this point, how many or what proportion of the craniosynostosis cases within 100,000 genomes do you think are now being diagnosed or have been diagnosed genetically? And do you have a figure on the proportion within GMS that are now achieving diagnoses?
So to date, and this is sort of fairly recent, we've analysed this, there's about 34% of patients within the 100,000 Genome Project that would be considered to have a genetic diagnosis. 5 of these have happened within the last 2 years, so since we published the paper in Genetics in Medicine. And these are sort of largely genes in which it's sort of required additional functional evidence to be confident that that variant has been pathogenic. In addition to that, there are a few genes in which they're sort of much harder to identify. So recently, a variant in one of the histone genes was identified as a de novo variant, but was originally flagged as having sort of bad reads in the VCF file when you sort of look at it. So ones like that, obviously, require intense scrutiny to look at in a bit further detail. It might be those sort of things that we're missing in future, and perhaps should focus our attention on.
All right, so just a couple of questions from the chat. First one from Verity Harthill, who asks about the families with variable penetrance variants and whether you've been able to image the unaffected individuals within families. Did you see anything in those?
I wouldn't have thought that they would have gone back and imaged the sort of unaffected for any sort of like the specifically unaffected individuals. We do tend to sort of see the families in units, sort of family unit at the clinics, in which they're sort of questioned about any other possible phenotypes. So largely we are aware if there's sort of any sort of subtle phenotype that might be evident, but I wouldn't have thought they would go through the sort of cranial imaging.
Okay, all right. And then the other one, I'm not sure whether you can see this question already from Nihal Joshi about the variants and transcription factors showing reduced penetrance, and does that help you from a therapeutic perspective? Not sure I get how that would, but that may be one that you need to address.
I mean, increasingly genes that we've found associated with craniosynostosis do display reduced penetrance. So obviously PRX1, which I've just presented, but also SMAD6, which is now the 7th most common cause. That is something we're sort of increasingly seeing. It's obviously very hard to explain why that's happening. But it's definitely something that we should sort of look at in future in terms of sort of potential new gene associations. I'd have to think about the sort of therapeutic point of view of it.
Okay.
But, yeah, I mean, just to say, sort of craniosynostosis, the current sort of therapy is surgery. So that's the sort of end point.
Okay, all right. Look, thank you very much for that, Rebecca. So we'll draw this to a conclusion then, and just remind you of a few dates. So firstly, the next seminar is on the 30th of May, also at 2 o'clock, last Tuesday in the month. That's our early career researchers event. And secondly, the Genomics England Research Summit, put 19th of September in your diaries. And if you can submit any abstracts by the 5th of May to events@genomicsengland.co.uk, that would be fantastic. With that, lastly, it leaves me to thank both of our presenters who gave really excellent presentation, and thank you also for answering all the questions. Have a good afternoon, everybody.